For example, some small molecules, such as haptens, they are antigenic in origin. Okay, so that means they can actually bind to the product of your B cells, or they can also bind to the surface receptors, but they are incapable by themselves of inducing a specific immune response. In other words, these substances, these small molecules that we call as haptens, they lack immunogenicity, okay, while retaining antigenicity, okay. This basically slide tries to explain it. Okay, so let's say you have, for example, this conjugate, which include hapten and a carrier protein. Okay, so as such, this blue portion, you would like to call it as carrier protein. For example, it can be BSA, it can be any other antigen that you are working with, any other immunogen that you are working with, right? And then you have these small molecules which are basically decorating this carrier substance or carrier protein. Okay. Now, if you have this conjugate and you inject in some form this whole thing to rabbits or any other species of vertebrates to induce an immune response, so you are going to generate antibodies, right? Now, those antibodies will be generated against what? Against, for example, number one, with these red or purple structures that you see on the surface, okay, which we call them as haptens. Okay, antibodies against haptens will be formed. You will also generate antibodies against these carrier proteins. Okay. Now, in addition to that, so you will also generate antibodies against conjugates. Conjugates means, so the specificity of this antibody will be against both the epitope coming from the carrier protein and some epitopes coming from the hapten molecule. So you will generate 3 types of antibodies. Right. Now, using this approach, actually the fine specificity of antibody was determined. So in that case, what they used was basically this dye or this small molecule that we call as dinitrophenol. Okay, dinitrophenol to nitrate group, and then you have this benzene ring, right? Now, hapten is basic. When you inject alone this dye, when this dye is injected alone, so you do not generate any antibodies. When you generate, for example, this dye, this small molecule, okay, along with a carrier protein, in this case BSA, you generate anti-BSA antibody, anti-DNP antibody, right? And obviously, if you inject only the protein, so you would generate anti-BSA antibody. Okay. So with hapten alone, you don't generate any antibodies. With carrier protein alone, you generate antibodies against the carrier protein. But when you mix this hapten and carrier protein, so you inject 3 types of antibody. Some of those antibodies generated will react to dinitrophenol, some will react to BSA, and some will react to DNP BSA combined. Okay, epitopes provided by both DNP and BSA. So now you know that hapten as such, it won't be able to induce these antibodies. Okay, but by this approach, for example, if you have generated this anti-DNP antibodies, right? And then you try to see whether in this sample, for example, if you collected the serum sample from these immunized rabbit, which were given heptin and this carrier protein, okay, and you want to find out whether or not anti-heptin antibodies are formed. So what you do is you just coat this DNP, dinitrophenol, onto the surface of these plastic plates, and then you come up with these antibodies, this sample, right? And then you would be able to detect that antibodies were formed against heptin. But if you inject heptin alone, you won't be able to detect these antibodies. So did you understand the difference here? If you have conjugated your small molecule with a bigger protein, it can then become immunogenic. Normally, it alone, it's not immunogenic. Okay. But nonetheless, the antibody generated against the anti-DNP can bind to this molecule as well. And this is what basically is the difference between antigenicity and immunogenicity. Okay, so actually the pioneer work in the field of antibodies and to determine the fine specificity of antibodies was performed by this guy, Carl Landsteiner. Okay. So what essentially he did was, so he used a similar approach that we described earlier. Okay. In this case, so this is one of the examples. So he injected, so basically this is the anti-sera generated. Okay. He used these different form of this dye. Okay. So you have aminobenzene, the other name is aniline. You have ortho aminobenzoic acid, you have meta aminobenzoic acid, and you have para aminobenzoic acid. Okay, so and by injection of this dye in animals, so he was able to get antibodies which were present in this anti-sera, and this anti-sera contained antibody against these different things. Now let's follow this table. What happened? Okay, so when this aminobenzene, it was able to actually induce, when this was injected, so it was able to induce antibodies against this dye, but not against subtle variation in this dye. So for example, if you have this orthoaminobenzoic acid, just by an additional group of this carboxylic acid was added, okay, so you didn't get any antibodies in the serum. Similarly, you didn't get anything against this metabenzoic acid and parabenzoic acid, right? When this orthoaminobenzoic acid was injected, antibodies were only obtained against this ortho form, orthoaminobenzoic acid, and not against this aminobenzene or with aminobenzoic acid or para-aminobenzoic acid. Okay, so you can actually follow this table just by changing the position of this group, change the specificity of antibodies generated. Okay, see, if you look at the composition more or less remains same. So you have this amino group and you have the carboxylic group. So you also have here amino group and carboxylic group. And same is the case here. It's just that position was changed. And with the change of position, you were not able to provide that epitope against which antibodies could be formed. So this work actually determined the fine specificity of antibodies. And from that time onwards, people started calling that antibodies bind to their antigenic moieties with pinpoint precision. Okay, very specifically they bind to these epitopes. Just by changing this position, removes the activity. Okay, rather the other contribution of Carl and Steiner, if you have gone through the historical perspective, basically the concept that we actually did not discuss, but I shared with you those 3-4 slides in which the contribution of different scientists was provided. So you would see that Carl and Steiner also got Nobel Prize in immunology. Okay, but he didn't get this Nobel for determining this fine specificity, and he himself recognized this being his pioneer contribution. Okay, so he thinks that if at all my contribution lies in the field of immunology, this is based on the antibody specificity. Just by this simple experiment where actually he used this dye in different formats, right? And he demonstrated the specificity of antibodies. But he was given Nobel Prize for discovering blood group antigens. So we know this ABO blood group. Okay, so that was actually shown to exist by Carl and Steiner, and that contribution was recognized for Nobel. All right, so just read this line. And Steiner's work demonstrated both the fine specificity of immune system for small structural variations on haptens and the enormous diversity of epitopes that the immune system is capable of recognizing. Okay, and this is the experiment he did for this.
Excuse me, sir.
Okay, so yeah. Go back
Sir,
one.
could you please go back to fine specificity of antibody slide?
This
Yes,
one?
yes,
Yeah.
sir. Sir, I just wanted to ask that from a clinical perspective, if we directly inject DNP in comparison to this hapten carrier conjugate, sir, what is the difference that we can find, and up to what extent we can get its advantage?
No, so you're saying that if you inject DNP alone,
Yes, sir, in comparison to the conjugate.
in comparison to the conjugate. So and then follow-up question was, so
Sir,
what would,
from a clinical perspective, what advantage does the conjugate have?
well, clinical will come later. First, it's basically how this recognition is happening, right? See, how do you activate your B cells? You activate your B cells by providing that antigen with the right specificity, which has the right specificity against your B cells. So you have your B cells, which has this receptor on surface of this, right? Now, if you come up with this hapten, this hapten has to bind to it, and then it will induce activation of these cells, and then antibody will start forming. Okay. Now, what I'm trying to say here is hapten alone is not immunogenic. It's not able to induce immune reactivity on its own. Okay. But many a time, so what happens is, for example, let's say you go to a physician and they invariably ask you, are you allergic to penicillin, for example, or some antibiotics, right? Only then they prescribe that antibiotic. And what is the purpose of that? Purpose of that is, these antibiotics are also small molecules. They are actually like something like as these small structures. And if you're allergic, so that means you must have been exposed to this earlier, and you must have induced some antibodies, or you might have some antibiotic reactive cells in your system. And whenever this antibiotic is injected, so you actually start developing something what we call as hypersensitivity reactions. Okay, which is very quick in induction. What I'm trying to say here is that hapten alone will not be able to activate your B cells, because it can bind to the specific receptor on surface, but the other signals which are required. So there are actually three signals which are very critical for induction of immune responses. One is this the specific recognition by the antigen. Second is the co-stimulations. Third is the cytokines produced in the milieu. So all these things, when are present, so you would be able to generate an immune response. Now, hapten alone would be able to bind to this antigen. Okay, but the other signals which are required, so those are missing. And when you inject this conjugate, hapten plus carrier, now how exactly it works, so we'll talk about it in little more detail. But for the time being, just remember that the carrier protein is processed by your APCs. They basically cut it into those small peptides. Those peptides are loaded onto MSC class 1, rather MSC class 2, to activate your T helper cells, and T helper cells can help provide those signal number 2 and signal number 3. And then you would be able to induce efficient antibody responses. So
Sir,
is that,
so here,
yeah,
sir, the message that you're trying to convey here is that all the three signals which are necessary for immunogenic response is delivered via conjugate and not
yes,
via DNP.
yeah,
Right, sir. Thank you.
yes, yeah. So hapten alone will not be able to provide all those signals. But if this is mixed with a larger protein, so we'll just talk about the characteristics of an immunogen. Okay, so then it will be a little more clear to you. So once you give this bigger protein, which is conjugated with the hapten, so then you can generate all kind of antibodies against the proteinaceous antigen as well as against the small molecule. So you have to have this complex form recognized by your B cells and T cells.
Right, sir.
All right. Yeah. So these are some of the factors which influence immunogenicity. Okay, so you actually can categorize them in different forms. So one is basically the intrinsic properties of an immunogen, and which are as follows. One is foreignness. Foreignness means what? To begin with, if you inject your own antigen in your system, so you will not generate immune response. Okay, because this is your self protein, and this is called self tolerance. Okay, your body usually does not induce immune response against your own antigens. However, if a foreign antigen is injected, so you start mounting an immune response. That's one. Second is molecular size. If you actually inject very small sequences of amino acid, so this is usually not immunogenic. Okay, so you have to have a certain size which can range from 1000 to 10,000 kilodaltons, and more complex is the structure, better its immunogenicity would be. Okay, smaller size structures are not immunogenic by itself. Okay, chemical composition and heterogeneity. Now, for example, if you have a homomer, for example, a polysaccharide, which has this single pattern, right? So you have this LPS, 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 right? Or only, for example, a repeating unit of that. You might have a bigger complex, a bigger antigen, but if the if the units are repeated multiple times, so it will not be immunogenic. Okay, so it has to have heterogeneity. For example, the other example I would give you is if you have a protein which has alanine, 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 which is not normally possible to actually make all these sequences, but you can artificially synthesize them. And once you don't have this heterogeneity, for example, alanine, proline, uh, aspartic acid, so all this heterogeneous nature of the amino acids have to be there in the protein so that it can be recognized as immunogenic by your B cells. Okay, if it has repeating units, it's not immunogenic. That's what it means by the third a property of an immunogen. Chemical composition, that means it has to be bigger in size, it has to be heterogeneous in nature, and repeating units are less immunogenic than if you have the heterogeneity in the representation. Fourth one is susceptibility to antigen processing and presentation machinery. Okay, now, so you have although we have not talked about the antigen processing and presentation, so we'll talk about these things at some time point. But if you inject a protein which is refractory to the antigen processing by your APCs, so you have this machinery that we call as proteasome. You also have this phagolysosomal degradation of your protein to generate those specific peptides. Okay, if those peptides are not generated, okay, from this antigen, so this will be less immunogenic. This rather will not be immunogenic. Okay, so your antigen has to be susceptible to antigenic processing and presentation. Okay, these are the intrinsic properties of an immunogen. Okay, now you can for example, the other example I think gave that if this product is inert, biologically inert, maybe you got a bone broken and then you got basically a rod to basically repair this breakage of bone. So this iron rod is not immunogenic. This is foreign in nature. Okay, but you will not be able to generate immune response against iron because this is actually inert, because it does not have this heterogeneity in chemical composition, and it cannot be degraded by your cells, so that those things can then be presented to your T cells or B cells, and eventually B cells and T cells can make the effector responses. So degradation is also very critical. So these are some of the intrinsic properties, right? Then you have the biological system. For example, genotype of a recipient animal is very critical. And we know that all of us have differences in our ability to recognize and respond to whatever we are injected with. Okay, when we talk about animals, for example, to generate antibodies, certain genetic background of animals are more able to induce immune response than the others. So the biological system also plays a critical role, an important role, if immunogenicity is to be induced or not induced. Okay. Now, the other thing is how much dose of this antigen you usually inject. Okay. And what route of administration you're following. Okay, so for example, you eat a lot of food, okay, through oral route, right? And usually you do not basically start generating response against whatever you eat. Otherwise, your survival will be very difficult. There are mechanisms how immune response is not generated against food items. So there are mechanisms in place. In your gut, you have something that we call as oral tolerance. if this is basically um, entering through these epithelial cells, okay, or enterocytes, so they actually induce tolerance. You don't generate immune response. But if you can specifically target it to, for example, M cells, which are present in your gut, so you can make uh, immune response. Okay, so many of these viruses, for example, poliovirus, so is usually processed through M cells, and eventually you can generate a good response even against an oral administration of this vaccine. Okay, but these things have to be optimized. You have to actually see what receptor is present on virus and what can be recognized by your the molecules present. The third thing that is also very critical is whether or not you are injecting this antigen alone or in combination with something that can efficiently induce immune responses. And this thing is called adjuvant. Okay, adjuvant. So usually if you inject antigen alone, you get very less immune response. If this is combined with something that we call as adjuvant, so there are different types of those, so you get a good immune response. And in these adjuvants, basic, they are nothing but the innate immune stimulants. Okay, so we'll talk about how exactly these adjuvants work. Okay, so this is basically the biological system. Some of the properties of biological system which contribute to the immunogenicity. And in this portion, so basically these are the intrinsic properties of the antigen or immunogen you are injecting. All right, so so let's first talk about the foreignness. Generally, if you inject something which has a greater phylogenetic distance between two species, it will be better in inducing immune response. Okay, so for example, if you get a protein from human X and inject in human Y, chances of it being immunogenic are very, very less. Okay, if you, for example, get this protein from horse and inject in humans, chances of it being immunogenic are more. Okay, if, for example, you get it from a fish and then inject in humans, so this will be even more immunogenic. And for example, if you get it from insects and you inject it in vertebrates, so this will be more immunogenic. So more is the phylogenetic distance between these two species where the source of antigen is basically where you have the antigen coming from and where the host species are involved. If these are distantly related, so antigen is going to be more immunogenic. And if they are closely related, this will be less immunogenic. Okay. Some macromolecules, for example, collagen and cytochrome C, they are more or less conserved through evolution. Okay. Through evolutionary process, if you look at the composition of collagen and cytochrome C, so they are more or less conserved from one species to the other species. And that is why it's not easy to recognize these antigens by your B cells and T cells. Okay, even if you get, for example, collagen from a rabbit and inject in your system, so it's going to be a little immunogenic. Okay, conservation is the key here. So one is phylogenetic relatedness. It has to be distantly related to be immunogenic. Okay, if the protein that you are injecting is conserved, so it will be less immunogenic. The third point is, if, for example, you have your own antigen injected, so they are going to be less immunogenic. Okay. However, there are exceptions here. One of them is, for example, corneal tissues or sperms. Okay. So because when your B cells and T cells were developing in bone marrow and thymus respectively, okay, so they somehow see a lot of antigen which you have, and if they see your own antigen, so they are deleted. But there are some antigen which they cannot really see. Okay, and because of this recognition which is missing during the developmental stage of your B cells and T cells, so you actually see these antigens as foreign. And examples are corneal tissues. So usually you have your cornea, right? And cornea is basically avascular in origin. Okay, so it does not have vascularization. So it is sequestered from your immune system. But somehow, if you got some injuries to your corneal tissues, um, so then you get actually a very robust immune response. Although corneal tissues are your own antigens, but during the developmental stage of your B cells and T cells, these antigens were not encountered. Okay. Uh, so apparently they are sequestered from your immune system. And because they were sequestered, so now they will be actually recognized as foreign antigens. So for example, let's say if you get this cadaver and basically get corneal tissues from this cadaver from a different individual, and you inject those, can make a single cell, rather you make a suspension of these corneal antigens and inject uh, even in the same individual. Okay, so you can actually generate immune response.
Excuse me, sir.
Yeah.
Sir, if there is any infection or any disease in the cornea, like you said, so sir, the immune system, how it will react, can we consider it as autoimmune reaction or not?
Well, many people do consider these recognition as autoreactive. Okay. Um, but initial cause actually is either injury or infection, right? But then you have this molecular spreading. Okay, or this is called molecular mimicry or epitope spreading. Okay. Uh, and because of that, so you can actually start inducing immune response to your own antigens which are present in corneal tissues, or even the components of the reproductive tract. Okay, so nervous system is another example of this. Normally, nervous system, corneal tissues, sperm, so these are called immunoprivileged sites. Okay, but if you get inflammation in these tissues because of injuries or because of other problems or infections, so then yes, chances of it being autoreactive in origin are high.
Right, sir.
Okay, molecular size. So usually, if antigen has a complex structure more than almost 100,000 kilodalton, 100,000 Dalton rather, rather 100 kilodalton, you can say, they are better immunogens. If the antigen has a molecular weight which is actually less than 10,000 Dalton, so these are poor in immunogenicity. Okay, so the more complex is the structure, better immunogenic would it be. More is the size, better immunogenic would it be. Okay, synthetic homopolymers, they are reduced in immunogenicity. Homopolymers. So that means you have repeating units, uh, many carbohydrates, right? So they are not immunogenic for the simple reason because it does not have that heterogeneity. Okay, so it just have those repeating units, and with repeating units, your immune system is not able to recognize it so well. The other thing is complexity in the structural organization. Okay, so you already know there are 4 levels of these protein structures. Okay, primary, secondary, tertiary, and quaternary. Quaternary structures would be much more immunogenic than your other structures. Primary structure rather are very, very less immunogenic in origin. Okay. Quaternary structure are the best immunogen. Okay, so proteins are the most, now if you basically compare the different biomolecules in terms of their ability to induce immune response, proteins are the most potent immunogens. Okay, after that you have polysaccharides. After that you have lipids, and then nucleic acids. Okay, um, and this is for the simple reason because your TCR and BCR, so they provide these structures that we call as rather paratope, so they can more efficiently recognize the counter structures present on these proteinaceous antigens than on polysaccharides, lipid, or nucleic acid. This is not to say that, for example, um, nucleic acid is not immunogenic at all. No, this is less immunogenic. Okay, so you must have heard about this disease, systemic lupus erythematosus, right? Where actually your DNA can be immunogenic in origin. Okay, but usually proteins are more immunogenic than any other kind of macromolecules that you have. All right, and if you actually provide these other units, for example, lipids, nucleic acid, polysaccharides, in complex with protein, so they will be immunogenic. Okay. Now, for T cell responses, only proteins and very few lipids serve as immunogen. Okay. As such, polysaccharides do not induce T cell responses. Okay, keep that in mind. So so you will know more about it as we go along with the antigen processing and presentation, right? But for T cell responses, proteinaceous antigens are the one which can activate your T cells. For B cells, you can actually generate antibody response against polysaccharides, lipid, and even nucleic acid to some extent. Susceptibility to antigen processing and presentation, which is very critical for activating your T cells, and T cells indirectly help your antibody forming B cells to form these antibodies, right? So, but susceptibility of this antigen to this machinery that you have in APCs is very critical parameter for inducing T cell responses. Okay, larger insoluble macromolecules are more potent immunogen than the soluble ones, because the larger molecules are more readily phagocytosed and processed. Okay, molecules which cannot be really degraded efficiently are less immunogenic. D-amino acid, for example, your system does not have the ability to degrade these D-amino acids. Okay, so and that is why they are actually poor immunogens. L-amino acids are, they can be efficiently degraded. Okay, biological system. We talked about these 3 things: genotype of the recipient animals, dose, route of administration, adjuvants. Okay, let's talk one by one. Dose and the route of administration. Now, if you inject too low a dose, you don't generate immune response. And same is the case with if you inject too high the dose of the antigen. This actually causes tolerance induction. This is called high dose tolerance, rather, right? Low dose tolerance and high dose tolerance. So you have to inject just the optimal dose if you want to generate good immune response. And this actually is in few micrograms. You don't require milligrams or grams or kilograms of thing injected. Okay, so just few micrograms is good enough for dose to be efficiently recognized by your immune cells. Okay, now repeated administration is always better. And why is it? This is basically the concept of induction of your rather formation of your memory cells and then elicitation of this response over a period of time. Okay, if you inject repeated doses, so you are going to get much better immune response as compared to if you are injected only once. And this is the concept with that you get this booster dose of vaccines and so on and so forth to get good immune response, right? Routes. You don't normally induce immune response when something is injected orally. And this is because of the oral tolerance phenomena. There are different types of cells which are present in your gut, which are actually tolerogenic in origin. How this antigen would be presented to your B cells and T cells is different in gut as compared to when this is injected intravenously or intraperitoneally or subcutaneously or intramuscularly. Okay, these are different routes which you can actually use. Intravenous, intradermal. Intradermal actually is always better. Subcutaneous is always better. Intramuscular is all right. IP and intravenous, you inject rarely. Okay, but orally you don't inject if you want to generate good immune response. And why is it? Because remember one of the slides I showed you that if you actually make a section of the skin and you stain with these antibodies which can detect the presence of APCs or dendritic cells. So basically you have a lot of those cells at these sites, intradermal route, subcutaneous or intramuscular route. So you find a lot of these APCs present there. And whenever you inject something, so this can efficiently start inducing your immune responses, right? So these are actually good route of immunization. Intradermal, subcutaneous or intramuscular to some extent. Okay, now they are saying that you can get this intranasal vaccination. Intranasal vaccination is not going to be that efficient if you have not really optimized your system. Okay, this is there are so many factors which decide the immunogenicity. Okay, adjuvant, again coming from this Latin word adjuvare, meaning help, to help. Okay, now substances that when mixed with an antigen and injected with it enhance the immunogenicity of the antigen, and this is called adjuvant. The key word is mixed with the antigen. So it's not like you have your skin, at one place you inject this adjuvant and another place you inject this antigen. You don't, nothing is going to happen. Okay, so this has to be first mixed, and then this mix needs to be injected if you want to generate good immune response, right? Effects of including antigens or rather adjuvants to induce immune responses. One of the mechanism by which these adjuvants enhance immune response is through persistence of antigen. Okay, So slow release of antigen at the injection site. So you have injected this mix of antigen with adjuvant, and over a period of time, so you will get the slow release of this antigen, and the slow release of antigen will induce efficiently your immune responses. One of the examples that you use is alum. Okay, this is the chemical form of this. Alum is actually a very commonly used adjuvant. The other thing, this adjuvant, what does it do is basically it provide co-stimulation. Co-stimulatory signals are enhanced if you include adjuvant in the immunization protocol. Okay, so it increases the expression of these molecules that we call as B71, B72, CD8080, another name on APCs, and their counter structure are usually present on T helper cells. And because of these interactions, so you induce good T cell responses. Okay, CD28 is expressed on, for example, T cells. CD8086 are present on your APCs, and when you have this interaction, so this is actually signal number 2. And that signal number 2 is efficiently induced if you include adjuvant in the cocktail. Okay. The third thing these adjuvants do is they actually increase local inflammation. At the site of injection, if you get an inflammation induced, so you can actually be rest assured that you are going to get a good immune response, right? If you don't get, if there is no swelling at the site of injection, if there is no inflammation, so chances are that something has gone wrong. The uptake of this vaccine or the immunogen is not efficient. Okay. Local inflammation. Fourth is non-specific proliferation of your lymphocyte is also provided by these adjuvants. Okay. Once you have ligated your specific B cells and T cells, then the environment induced by adjuvants can non-specifically keep on increasing the number of your T and B cells. Okay. For example, granuloma formation is one of the key factors to induce a good immune response. Okay. Although these are not used for human medicine these days. Incomplete adjuvants and fluent complete adjuvants are the chemicals which serve as adjuvant, and they can actually very efficiently induce the immunogenicity of any antigen. And how do they work is by attracting a lot of these macrophages at the site of injection. And you get this indurated structure formed at the site of injection. This is called granuloma. Okay, so any clarification on this slide? So the next part, the epitope, we are going to talk about in next class.
Sir,
Yeah.
so in the fourth point, you said that this stimulates more amount of non-specific proliferation, but why do we require like non-specific? Because specific lymphocytes will actually like get attached to these antigens and induce the response. So like, what is the importance of non-specificity?
Non-specific means, so the components of adjuvant are not going to stimulate your specific cells. But once the specific cells are formed, because you are already including the antigen, right? So now, because of these adjuvants, so you have produced these cytokines at the inflammatory site, and these cytokines can also be then they can go to lymph node and in the draining lymph node, and because of their growth promoting activity, so the T cells which were induced to begin with by your antigens now they can keep on expanding in number, and as these cells expand in number, so more and more B cells will start making antibodies, and more T cells will start getting formed, right? So this is that non-specificity means. Non-specific stimulation of your lymphocytes. And many a time, the stuff which is induced following injection of these adjuvants, they can actually also serve as what we call as mitogenic, rather they can actually promote mitogenic response. And mitogenic is basically you just promote, you induce mitosis of your B cells and T cells. Okay, so essentially, so your cells are already activated, and now they can actually get in cell cycle more quickly.
Fine, sir. And so another point that in the last class you had discussed about the images of facts, like flow cytometry, basically. So during that, in the third image, you asked a question basically like in the fourth quadrant, what are the cells signifying? So while answering that, actually your voice was broken, so we couldn't hear. So could you just repeat that point? Whether they are cytotoxic T cells or not? So this
This one?
this one, sir, the other one, like fourth quadrant. So
this one? Or
no,
this
so
one?
below one. Yeah, this one. You asked that what are
Well,
these?
yeah.
So.
No, I think I answered. Rather, you answered that question, right? And this is right, that these actually are cytotoxic T cells. These are helper cells.
Fine, sir.
Yeah. Okay, so any more questions if you have, so we can take those maybe in, what time is it? Well, it's all time already. Okay, so we are going to stop it now. And I will upload these slides on the Moodle page, and you can go through them. And your questions will be taken up on Mondays, in Monday's class. Are adjuvant immunogenic by themselves? Well, adjuvants alone are not immunogenic. Okay, immunogenic in the sense they can actually activate your innate immune cells. But for example, whether you can induce immune response against these oils components, so no. Okay, so the right answer is no. Okay, they can stimulate innate cells, but not specific cells. Anyways, so we are going to stop it here.